Dear students, welcome to the calculus class. In this video, we are going to study about oxygen transport. Oxygen is transported in two forms. One is in the form of oxyhemoglobin. Second is in dissolved state in plasma. In the form of oxyhemoglobin, 97% of oxygen is transported to the tissue. In a dissolved state, only 3% of oxygen is transported. Our tissues require 250 ml of oxygen per minute. This is the consumption rate of oxygen by the tissues when the person is at a rest. But in dissolved state, very less oxygen is transported. Only 3% is transported. That is, per liter, about 3 ml of oxygen is transported to the tissue. If our cardiac output is 5 liters per minute, then only 15 milliliters of oxygen is given to the tissue. But the actual demand is 250 milliliters per minute. So, to meet this demand, oxygen is transported in the form of oxyhemoglobin. Oxygen binds with the hemoglobin and forms oxyhemoglobin. In this form, 97% of oxygen is transported. 1 gram of hemoglobin carries 1.34 ml of oxygen when it is 100% saturated. 100 ml of blood on average contains 15 grams of hemoglobin. So the 100 ml of blood transports 1.34 ml of oxygen into 15 grams of hemoglobin. This is equal to 20.1 milliliters. 100 ml of blood transports 20.1 milliliters. If cardiac output is 5 liters, how much? Let us see. 1 liter blood carries about 20.1 into 10. This is equal to 201 ml of oxygen. If a cardiac output is 5 liters, 201 into 5 liters. This is equal to 1005 ml. But the requirement is 250 milliliters per minute. Our blood supplies 1005 ml of oxygen. So, it is more than the requirement of oxygen to the tissue. That's why most of the oxygen is transported in the form of oxyhemoglobin. 97% of oxygen is transported in the form of oxyhemoglobin and only 3% is transported in a dissolved state, dissolved in plasma. So, how oxyhemoglobin forms, let us see. For understanding it, we have to know the structure of the hemoglobin. This is one hemoglobin. Hemoglobin has 4 polypeptide chains, 2 alpha chains, alpha 1, alpha 2, and 2 beta chains. This is the beta 1 and beta 2. So, total 2 subunits are there, alpha subunit and beta subunit. Each subunit is a dimeric, means 2 polypeptide chains are present. So, it has total 4 polypeptide chains. Each polypeptide chain has one iron containing heme group. So, thus 4 heme groups are present in hemoglobin molecule. Each heme group is capable to bind one oxygen molecule. So, how many heme groups here? Total 4. 1, 2, 3, 4. So, 4 oxygen molecules can bind to this heme group. Thus, they form oxyhemoglobin. When 4 oxygen molecules are attached to the hemoglobin, then we say that hemoglobin is 100% saturated. Now, let us take this analogy. Here, this is the hemoglobin. This is the blood plasma. This hemoglobin is equivalent to a car. Let us assume that in this car, 4 seats are there. Each seat is one heme group. 4 persons can sit on these seats. These persons are oxygen molecules. When this car is occupied by the 4 persons, it is 100% filled. In the same way, when the hemoglobin is bound to 4 oxygen molecules, then we say that 100% saturation hemoglobin got. And this is the oxygen dissolved in the blood plasma. How this oxyhemoglobin forms? To the hemoglobin, oxygen binds with a weak bond. When oxygen binds to hemoglobin by a weak bond, then this phenomenon is called oxygenation, not oxidation. This is oxygenation. It is a reversible reaction. Hemoglobin and oxygen bind and form oxyhemoglobin. That is called association. When oxyhemoglobin separate, they form hemoglobin and oxygen. This is called dissociation. The formation of oxyhemoglobin is called oxygenation, not oxidation. This is the hemoglobin. Hemoglobin shows very less affinity towards the oxygen. Even though it has 4 hemes for binding the oxygen, it is very least interested to bind with the oxygen. Okay, this is the car. In this one, 4 seats are vacant. Now, the driver comes and sits in the car. When oxygen binds with the heme group, structural changes occur in the hemoglobin. The structural changes are called conformational changes. These conformational changes increase the affinity of hemoglobin towards the oxygen. So, the second oxygen molecule also goes and binds with it. And this binding further increases the affinity of the hemoglobin towards the oxygen. So, the third molecule also binds with it. Now, it is 75% saturated and only one position is left out. It will show more affinity towards the oxygen than the previous places. So, it binds with the oxygen. Thus, hemoglobin is saturated, 100% saturated. These changes, the loading of hemoglobin occurs in the alveolar capillaries. That is the boarding point of oxygen. This boarding or this loading of oxygen depends on partial pressure of oxygen in the alveolar blood. Tissues are equivalent to alighting points. Here oxygen partial pressure is less in the blood plasma and in the tissue capillaries. So, from the hemoglobin, the oxygen is delivered to the tissue. First, oxygen is released into the blood plasma. From there, it goes to tissues. Dissociation of oxyhemoglobin is taking place. Oxyhemoglobin is breaking up and forming hemoglobin and oxygen. Now we call this hemoglobin as deoxygenated hemoglobin, and that blood is called deoxygenated blood. Though it is called deoxygenated blood, it contains oxygen. Now this blood from tissues move to the alveolar capillaries. Now deoxygenated blood along with the hemoglobin moves to the alveolar capillaries. That is the boarding point. Due to the high partial pressure of oxygen in the blood plasma or in the alveolar capillaries, and also here only one place is vacant. The high affinity of hemoglobin towards the oxygen easily binds with this hemoglobin. Now it is saturated, 100% saturated. From here it goes to the tissues and delivers the oxygen. The formation of oxyhemoglobin and dissociation of oxyhemoglobin depends on partial pressure of oxygen in the blood, that is in blood plasma. Let us find out the relation between the partial pressure of oxygen and percent oxygen saturation. Percent oxygen saturation is the percentage of oxygen Saturated oxygen percentage. When a graph is drawn between the partial pressure of oxygen in blood plasma, this is in blood plasma, and percent oxygen saturation, a curve forms that is called oxygen dissociation curve. This is the oxygen dissociation curve. I'll write here the graph drawn by taking the partial pressure of oxygen and percent of oxygen saturation is called oxygen dissociation curve. This oxygen dissociation curve is a sigmoid curve, S-shaped curve. In this curve, we can find two parts. One is the flat region, plateau region, and it indicates the hemoglobin saturation at alveolar capillaries. Next, this is steep. The steep indicates the loading points, and this the plateau indicates the after the loading in alveolar capillaries. If you see this graph, at 100 millimeter mercury, the percent oxygen saturation is only 98 or 98.5. At 60 millimeter mercury, it is showing 90% saturation. At 100 millimeter mercury, it is showing only 98% saturation. So 100 to 60, this variation, even though 40 millimeter mercury difference is there is no much difference in the saturation of oxyhemoglobin. At 40 millimeter mercury, it is showing 75% saturation. Means with 20 millimeter mercury difference, there is a lot of change we can observe in the saturation of hemoglobin. Between 60 millimeter mercury and 40 millimeter mercury, if we take, the difference is 15%. When we take the difference between the 100 millimeter mercury and 60 millimeter mercury, only 8% difference is seen. At 100 millimeter mercury, 98% of saturation we can find. At 60 millimeter mercury, the saturation is about 90%. So only 8% difference we can find between these two. At 40 millimeter mercury partial pressure of oxygen, the percent oxygen saturation is 75. That is 75% saturation is observed. Here, in the partial pressures, the difference is only 20. But in the saturation, we can see the 15% of difference between these 2 points. At a tissue level, the partial pressure of oxygen is maintained around 40 millimeter mercury. So, easily oxyhemoglobin dissociates and oxygen is delivered to the tissue. Every 100 ml of blood delivers 5 ml of oxygen. Now look this one. This is the 50% saturation. 50% oxygen saturation. At 50% oxygen saturation, the partial pressure is around 25. This is 30. This is the 25. The partial pressure of oxygen at 50% oxygen saturation is called P50 value. P50. P50 is how much here? 25. This is oxygen dissociation curve. Oxygen dissociation curve is a sigmoid curve. It has one steep area and one flattened or plateau. Plateau lies between 60 millimeter mercury to 100 millimeter mercury. Between 0 to 60, the steep is present. Here, this plateau indicates that 4 oxygen molecules are bound to the hemoglobin. At 40 millimeter mercury, only 3 oxygen molecules are bound to the hemoglobin. Now, we will compare it with the fetal hemoglobin. This white curve shown is the fetal hemoglobin. This pink one is the adult hemoglobin. Fetal hemoglobin shows more affinity towards the oxygen than the maternal hemoglobin, because in maternal hemoglobin, the polypeptide chains are 2 types. That is, the 2 subunits of polypeptide chains are there: alpha subunit, beta subunit. Here alpha 1, alpha 2, and beta 1 and beta 2. These are present in the maternal hemoglobin. In the fetal hemoglobin, it also contains two subunits. One is the alpha 1 and alpha 2. In the place of beta subunit, gamma subunit is there, gamma 1 and gamma 2. The polypeptide chain structure of the gamma or the structure of gamma chain shows more affinity to the hemoglobin. So, fetal hemoglobin shows more affinity to the oxygen than the maternal hemoglobin. That's why in the capillaries of maternal side placenta, oxygen partial pressure is even though low, that is 35 to 50 millimeter mercury. The fetal hemoglobin is capable to bind with that oxygen. In the maternal hemoglobin, P50, this is the 50% saturation, P50 is 25. But in the fetal hemoglobin, it is 18.5 to 19. So, P50 value is very less for the fetal hemoglobin, and P50 value is more for the maternal hemoglobin. In maternal hemoglobin, P50 is 25 millimeter mercury. For fetal hemoglobin, P50 is 18.5 or 19. So, when the P50 value is less means the affinity is more. Now the factors affecting the oxygen dissociation curve. One is the temperature. Second one is CO2 concentration. And third is hydrogen ion concentration. And fourth one is BPG, bis phosphoglycerate. Assume that this side high temperature, carbon dioxide concentration, hydrogen ion concentration and bis phosphoglycerate concentration bring changes in the sigmoid curve or in the oxygen dissociation curve. When temperature is very high, carbon dioxide concentration is very high, hydrogen ion concentration is very high. When the BPG is very high, then the dissociation of oxyhemoglobin occurs. Means at this time, oxyhemoglobin dissociates into hemoglobin and oxygen. This side let us see low temperature, in low temperature and low carbon dioxide concentration and low hydrogen ion concentration and 4 low BPG. In these conditions, this graph moves to the left to the normal curve. So it shows that at a low temperature and carbon dioxide concentration is less, low hydrogen concentration is nothing but high pH. High hydrogen concentration is low pH. And when the BPG is less, then it will shift to left side. And the graph shifts to right when the temperature is high, carbon dioxide concentration is more, and hydrogen concentration is more, BPG is more. In these conditions, the graph shifts to right. The effect of hydrogen ion concentration on the formation of oxyhemoglobin or percent oxygen saturation is called Bohr's effect. In Bohr's effect, the curve moves from left to right. Reverse to that one means when the temperature is less and carbon dioxide concentration is less, hydrogen concentration is less and pH is more and BPG is less, at that time this curve moves to left side, this side. When the temperature is more, the graph shifts to right. When CO2 concentration is more, then Bohr's effect is caused. One more. When the oxygen concentration or pressure, partial pressure is less, at that time also Bohr's effect is caused. When the hydrogen ions concentration is high, Bohr's effect is caused. pH is less, Bohr's effect is caused. When the BPG is more, then Bohr's effect is caused. Thank you for watching the chocolates class.